Thanks for coming to Grounds on the Go. This is a seminar series that my office, the Applied Research Institute at the University of Virginia, co-sponsors with the UVA Research Park in coordination with Katie Murphy. And we put together this series to bring researchers from the University of Virginia up to the park and into the community so you can learn about the research that they're doing. And so today, I'm very excited to have my friend from the Department of Biology, Sarah Kucenas, here to tell you about her really interesting work using these amazing little fish. So I want to introduce Sarah. Sarah Kucenas is Associate Professor of Biology, Cell Biology, and Neuroscience. And she is interested in elucidating the cellular and molecular mechanisms that mediate glial-glial interactions in the developing and regenerating nervous system. Her long-term goal is to understand the mechanisms that mediate cellular interactions along developing spinal motor nerves and use this information to better understand how the human nervous system is initially established, maintains, and then behaves during diseases. Sarah earned her BS in biology from Valparaiso University in 2000, and then went on to earn a PhD in pharmacological and physiological science at St. Louis University with Dr. Mark Voigt in 2005. And after Dr. Kucenas' postdoctoral work with Dr. Bruce Appel at Vanderbilt University, she joined the faculty at the University of Virginia in 2009. Sarah is married with a 6-year-old daughter, Madeline, who's adorable, and has 2 dogs and is an avid swimmer. And so thank you so much. Please welcome Sarah.
Thank you. So can everybody hear me? Okay. I'm really excited to be here today to talk to you about 2 things that are really kind of near and dear to my heart. The first is this wonderful model organism, Zebrafish, and I'm going to give you what I think is a pretty cool background into this model organism, its history, why we use it in biomedical research, and then spend maybe the last half of our time together today showing you how my lab uses it, where we're particularly interested, as Melissa said, in understanding the nervous system. And I'm going to try to keep this very kind of 30,000-foot view. But if you have questions in the middle, I teach, so I don't mind if people want to stop me in the middle and ask questions. I can ask them at the end. I'm not going to go into a lot of detail initially, but please ask me more specific questions if you have them. So I wanted to kind of step back for people in the audience maybe that aren't biomedical researchers and really kind of define what a model organism is. And so as a biomedical researcher, when I'm interested in studying the human nervous system in terms of development and disease, I can't necessarily study humans. And there's many reasons for that. Historically, there are laws against that, but also practically, right? We can't see how the nervous system develops in utero, which is when the majority of the things that happen that create kind of the brain and spinal cord and the structures that we need in our nervous system are developing is during that first 9 months when a woman is pregnant. And so we use model organisms to kind of as a proxy to understand how these things happen. And so a definition of a model organism, just kind of so we're all on the same page, and I apologize if this is small so I can read it to you. It's a species that's widely studied, okay, so that's an important component, usually because it's either easy to maintain or very easy to breed in a laboratory setting. It has particular experimental advantages, which I'll talk about today, and these are always non-human species, and they're used to help scientists understand biological processes. And so model organisms are quite varied. I'm a biomedical scientist, and so for me, model organisms that have attributes that allow me to conduct experiments that make me think about humans is important. But it's also important to know that a lot of biomedical research, or scientific research in general, is really meant to just ask fundamental questions on why things happen. Just very cool phenomena that occur in our environment, why life exists. And so today I'm going to focus more on the biomedical side because that's my interest, but I kind of want you all to appreciate that organisms can be used to study just those questions of why in life on Earth. And so why we use them in particular, as I said, if they can breed in large numbers, that's really, really helpful. When we think about human disease, right, it's a one. Most often, you have one offspring at a time. You may have twins or multiples, but when you want to study something kind of very robustly, we need to have organisms that create lots of progeny so that we have many kind of reproducible units that we can study. What's ideal is if they have a short generation time. So again, as a human, right, when you're interested in nervous system development, actually, the nervous system doesn't stop developing until you're approximately 25 years old. Okay, it seems like a bit of a shock, but as an undergraduate educator, we know that that's true. The nervous system is not done until they're kind of into their adult stages. But I don't have 25 years to study some of these nervous system phenomenon. And so having model organisms that I can see development occur very rapidly is important. They need to have genes that are similar to humans. So genes are kind of that blueprint, if you will, that exists in all of our cells. And it's that genetic material that kind of drives development of all of the organs in our body, including the nervous system. And so having a model organism that is genetically similar to humans is very powerful, because then by studying gene X in zebrafish, I'm more likely to be able to kind of attribute those functions to gene X in humans. The ability to study mutants, and I know it sounds like a bit of a weird word, but that is actually the word we use in science. Being able to create mutations in that genetic material, in those genes that are similar to humans, and study what function those genes have is very important. Because what we know is that the vast majority of disorders, probably all, have genetic bases, which means there's a mutation or a defect in kind of the coding of a gene in our genome that causes a human disease. And if we can model that by mutating, creating a mutant in that same gene in zebrafish, we can actually study it in a model of an organism. We really like model organisms, and I'll talk to you about zebrafish with this, where we're able to test new drug targets, right? So using model organisms is not just important in terms of being able to kind of create a model of a human disease, but being able to then take that model and treat it with a drug to see, can we fix that disease? As a proxy, it's kind of what we consider preclinical testing. Let's test if these drugs help the fish model of disease X, and then if it does, we can kind of extrapolate and start to ask, can we then treat this human patient with the same kind of disorder with this gene? What's intriguing for people like me and many people that study development of the nervous system is having a model organism that actually allows us to study development. So as I mentioned earlier, humans, most of the nervous system development that occurs, it's actually the most important, is in the 9 months that a woman is pregnant. We can't see that. Those same stages of nervous system development occur in mice. However, a mouse mom is also developing an embryo in utero, so we can't watch it. But if we can have a model organism where that development occurs where we can see it outside of the mother, that actually allows us to study these events that we can't possibly kind of look at in humans in a model organism. And finally, what I think is important for a lot of people to appreciate, but they don't maybe necessarily know, is that a lot of animal research actually eventually leads to treatments for animals as well. So it's not that just we're using animals as a proxy to treat humans ultimately. There's many, many examples of diseases that occur in both, for example, dogs and humans. Duchenne muscular dystrophy is one of those diseases. Duchenne's is a muscular dystrophy that affects young boys. Dogs have the exact same disorder. And so studies that look at Duchenne muscular dystrophy in zebrafish and mice and dogs are leading to treatments not only for humans, but also, maybe not fish, but dogs, right? So this research is very important both biologically in terms of a scientist, but also it's not just that we're using the animals and they're not getting any benefit. So this is a slide of some common model organisms. This here is a mouse. This is probably the most common model organism when you think about biomedical research. They're incredibly powerful because they're vertebrates. So they have very similar kind of evolutionary trajectory through time that we do. They have all the same organ systems. They develop in exactly the same way, and they're mammals, and they look like us. And so when we think about studying diseases, if the animal looks like us, we just kind of subconsciously think that what we learn in that organism probably is going to be the same in us. However, hopefully what you can appreciate is that although this is a cute little organism, if I'm a nervous system developmental biologist, it's going to be very difficult for me to look at the nervous system in these guys. Okay? Another really common model organism are frogs, believe it or not, this African clawed frog. It has some benefits that the mouse does not. So as I mentioned, embryos develop in utero in the mouse. And so when you think about developmental studies, you can't really get at them in a mouse. But frogs lay their eggs. And so we have the ability to look at development occurring in these organisms. But unlike zebrafish, these eggs of these frogs are not see-through. They're not transparent. And so it's very difficult, even though the egg is developing in front of us without sort of the obstruction of an abdomen or some sort of uterine wall, it's very difficult to see these developmental events. These are some others. Drosophila, this is your common fruit fly. So if you've gone to Carter's Mountain and you've gotten a bag of apples, these guys come flying out of that bag. Believe it or not, these guys are used for biomedical research. Although they're invertebrates and they're flies, they have really complex behaviors. And so people use Drosophila to study the nervous system because they have all of the same cell types, they use all of the same genes, and they have very complex social and behavioral outputs. And they're a little bit more tractable than a mouse, okay? This is a worm. Although you can see through it, hopefully you can appreciate there's not much complexity there. So there's this trade-off of having organisms that are simple, that allow us to see things that are happening, but that have enough complexity that we feel like that we're going to learn something that's attributed to a human disease. This is yeast, and this is cell culture, which is another very powerful technique. And a lot of people always ask me, well, why don't you just study human cells? Right? Why don't you take these cells out of a human and put them in a dish? You can see them. They're easy. You can get lots of them. And then you can do whatever you want, right? The problem with that is we've gone from incredible complexity to absolutely no complexity. And so although we can study human cells in a dish, there's no environment that these cells exist in that is anything like the human brain, for example. And so what we need, and what somebody thought we needed a few years ago, and I'll give you the history, is another model organism, okay, to be able to kind of really ask questions about human development and disease, in particular with the nervous system. And so I just wanted to put these kind of more graphically into a view for you, for those of you that are not as common, as kind of well attributed to the animals. This is an overly simplified evolutionary tree, and typically trees are not shown this way, but I think this is the way to get the point across, is that the first primordial life starts here at the bottom of this tree, and that what ends up happening is we had diversification through time. So as you go up the tree, you're going through what's called evolutionary time, and you get more diversity, you get more complexity in life. And so here is fungi, so these are things like yeast and other fungi. You then get the plant branch. You have invertebrates, so this branch would have those fruit flies and those worms I told you about. But it's this animal branch here that goes off into the vertebrate lineage where we see things like the fish, the frog are here, you're going to have mouse and people, okay? So even though there's a significant, it seems like this branch is quite long, we're actually more closely related to things like frogs and fish than we are to some of these other model organisms. And in terms of time, I didn't appreciate you all would be so far away, I apologize. We are about 300 million years removed from all of these model organisms. Now that seems like a significant amount of time, but actually when we think about our kind of link to these organisms as all being vertebrates, that means that our developmental patterning is the same. Even though we're 300 million years apart, it's not that we've changed dramatically in terms of our building blocks. The cells are the same, the genes are the same, the organ systems are the same. What's changed is the complexity of the systems. And so you're saying to yourself, you just spent 5 minutes telling me about all these other wonderful organisms. Why zebrafish, right? Why did somebody think we needed another model organism? And so I wanted to give you a little bit of a historical perspective at the time that the zebrafish was being developed. In 1953 was the discovery of the double helix, the structure of DNA. This was a big deal. This wasn't that long ago. And so you have to appreciate that although we knew that there was genetic material in cells, by being able to appreciate how that information was structured, it immediately allowed for the creation of this dogma, which holds true today, which is called the central dogma of biology. And it's this idea that we know every cell in our body and every cell in any living organism has this DNA, these genes. It's the blueprints, right, of everything that we are. And every cell has the exact same blueprint. But what's different is that which genes are turned on at what time, and that's what helps us diversify. That's what helps us make a heart cell, for example, versus a cell that's in your brain. And so during this period, this 10 or 15-year period, we figured out the structure of DNA. We figured out very rapidly then that that structure was heritable. It could be passed between people. We started studying heritable diseases. We started to understand how life existed on Earth in a really powerful way. But we didn't have any model organisms that were really easily tractable, and that's where this guy, George Streisiger, came in. He was a phage geneticist at the University of Oregon. And so he had spent his entire career studying mutations in DNA and how mutations in DNA could be passed from generation to generation, and the importance of these mutants, right, quote unquote mutants, in creating kind of disease phenotypes. But he was getting kind of frustrated because this really wasn't where biology was going. We were moving past the fact that you could make a mutation and that it was heritable. The question is, what did these mutations do, right? How did they contribute to human disease and development? And so why, though, does he need another model organism? Why didn't he just jump into some of these other ones and use this newfound ideas of central dogma and DNA to drive biomedical research? Well, as I mentioned, the mouse is great, but you can't see through it. There's not a lot of easy ways to get at nervous system development. And so this was not the best organism. Yeast, although an incredibly powerful model when thinking about genetics, there's nothing that looks like a nervous system. There is no nervous system in yeast. And so simply by understanding the structure of a gene was not going to tell him anything about the nervous system in a human. Frogs, as I mentioned, you can see through them in the sense that they're laid, right? There's no development in utero, but the eggs are very muddy. So there was not a real easy way to see what was going on. And then, as I mentioned, cell culture, you put a human neuron, and I'll explain what that is in a little bit, into a dish. It doesn't at all behave the same way as it does in an actual human brain. And so he was particularly interested in development and development of the nervous system. And during this time, he had a little bit of competition. Okay? There was this guy, Seymour Benzer, famous Drosophila geneticist, who was using these fruit flies to study complex behaviors. And at the same time, there was this guy, Sidney Brenner, who won the Nobel Prize, using worms, although a very simple model, looking at the nervous system. But what Streisinger said is, these are great models for the questions they're asking, but the brains, the nervous systems of these organisms look nothing like a human's. So you're going to learn some fundamental things, but they're not going to tell us a lot about complex behaviors that occur in human beings or complex diseases. And so Streisinger took a different approach. This is a very different time when he was writing grants. He actually wrote a grant to propose an experiment. He didn't actually have to give data to get the money. And so this is a quote from a grant he wrote to the NSF. He wanted to propose to use an organism. He didn't say zebrafish yet, but he was asking for money to figure out how to do this, to study the organization and development of the vertebrate nervous system. Okay? And he wanted to develop a novel organism to allow this to occur. And so where we are now is zebrafish. So there's an interesting story aside. So people say, how did Streisinger come across zebrafish? Why, out of all the fish on the planet, did he decide that this Danio rerio was the right model? So it turns out there's a lot of history now about Streisinger since he's passed away. He was an avid tropical fish collector as a child. So apparently he spent most of his young and adult life visiting aquariums everywhere his parents went. He had really large aquariums at his parents' house, and he collected all of these different tropical fish because he was fascinated in them. And that probably is why he circled back to them, because he understood their life cycle. He understood the ease of breeding them. He understood how easily he could collect their eggs. And he focused in on this zebrafish, this fish known as zebrafish. So it's a tropical fish, right? But it's freshwater. So they're very easy to maintain. You can keep them in normal water. You just have to heat it up a little bit. There were other systems he tried, but these guys were the hardiest. And so if you've ever had a fish tank as a child, you probably had a Danio rerio or a zebrafish in it. You can go to Walmart nowadays and get them for $0.25, right? My lab always asks me why we don't just go to Walmart to get the fish, because the fish we get are probably $200 or $300 a pair. They're bred to be research animals, obviously, right? But he started with pet store-owned fish and tried to develop these guys as a model organism. And so why were they the best model? Okay? What's great about these fish is that you can put them in these what we call nuptial chambers in a lab setting where you put males and females together at night. When the sun rises or the lights come on in my facility, that triggers the behavior to start to breed. And so within 5 minutes of the lights going on every morning in my facility, the males and females that are put together start laying eggs. And we can put them in these tanks that have these mesh bottoms so that when the eggs are laid, hopefully you can appreciate these little yellow things, which are collected here, they've laid their fertilized eggs. And so very, very easily, every morning, we can go in and get eggs. I have about 15,000 adults in my facility. We don't cross them all every night. But you can imagine we have thousands of embryos that we can use. And that gets back to that idea of what's important in a model organism. Easily bred and lots of them. Okay? So here's a dish of collected eggs. That's what they look like, little pearls in the water. And the reason they're the best model organism among many, and I'll keep talking about them so you'll all be convinced, but like the frog, but better, they are truly transparent. So they're laid. There's no in utero development, so there's no kind of worry of obstruction. But the frog is very muddy, whereas the zebrafish embryo is not. And additionally, in addition to that, these embryos develop very rapidly. So what you're watching here is a single cell embryo that was fertilized, and you're seeing those very early cleavage events of that single cell. This looks exactly like a human egg that's been fertilized by a human sperm in a dish. This development is identical, but the difference is that you start to see these processes that are going to create the head of the fish. You'll see an eye here and a tail. The brain is forming here. Here's the body. And then eventually this organism, if this movie were to continue, this would become a zebrafish. Okay? This movie, although sped up, this stage, going from single cell to the stage right here, takes 20 hours. This same stage in a human would take 4 to 6 weeks. And so the ability to get this animal to develop at this speed very, very rapidly, plus the fact that we can get thousands every morning, allow us to ask really robust questions in science. And then this is just to give you a perspective. We have staging charts. So what I showed you was that movie to about this stage. It's about 18 to 20 hours. But by 3 days, so if my lab crossed on Monday, by tomorrow morning, they'll have fish that are 72 hours. Their eyes are fully functional. The fish are swimming. They have complex behaviors. They have working organ systems. And they're ready to be bred and grown into adults. So within 3 days, we have a fully developed organism that's at about the same stage as an 8-year-old human. And that's pretty powerful when we're thinking about wanting to really study nervous system development and disease. The other thing that I really want to kind of drive home, and this is a slide I show my undergraduates, is the reason we use animal models to study development, especially vertebrates, is because it does allow us to directly kind of correlate things that we're finding in these organisms with those in humans. And so this is a slide. There are 7 different organisms here, and this is known as the phylotypic stage of development. You have to pardon me, I'm a teacher, so this is the phylotypic stage of development. And so these are 7 different organisms, and in vertebrate development, all organisms develop exactly the same up until this point. Does anybody want to take a guess as to which one is the human?
6.
Number 6, okay, anybody else? Yeah? 4? None of them. Well, one of them is, I promise. I wouldn't do that to you. Okay, well so we're going to go just a little bit further. Oh, and let me say, this is about 4 to 6 weeks of development. For one of these that's the human is about 4 to 6 weeks of development. The one that's a zebrafish is only about 18 to 20 hours, so keep that in mind. Okay, we're going to go to the next stage. It's going to get easier. Okay, so you could tell that these two are probably going to become something like a fish or amphibian, right? Probably 7. Okay, yes, you're right. But what I think is important, and this is, it gives it kind of a pictorial view, is that being able to study an organism like zebrafish, which is kind of number 1, and these earliest stages that build a human nervous system, whatever we learn there is going to be directly translated to our understanding of human development. Now, obviously, you don't study lungs in something like a fish, right? But in terms of the nervous system, what we're finding is that the development is nearly identical. They're also genetically similar, which I told you is very important. Because if we're going to study, let's say, a gene that we know is important in the nervous system, we want to understand or appreciate that that gene is going to also probably play that same role in humans. And so more than 70% of human genes are found in the zebrafish genome. So they're more than 70% identical genetically. And 84% of human disease-causing genes that we know are found in zebrafish. Okay? That's incredibly powerful. That means 84% of diseases in humans we can study directly in fish by mutating or affecting that gene and understanding development. This is an example. You don't need to necessarily read all the details, but I wanted to give you a real-world example. So 16p11.2 is a deletion. It's a common deletion, not common, but it's a deletion that's found in human patients. Deletion of this chromosomal segment leads to developmental delays, intellectual disability, different types of autism. These children often have trouble with socialization and language. And so this huge deletion in humans, which gets rid of all of these genes because it's essentially just a big chunk of the chromosome where that genetic material is removed. And it's very difficult to understand what in that region is kind of causing these phenotypes that these kids have. But if we look at zebrafish, which is here on the right, even though zebrafish don't have all of these genes on one chromosome or one piece that was deleted, the power of the fish is that the vast majority of these genes have counterparts. That's what the red lines kind of show you. But instead of deleting the entire region in humans, where we don't know, is it one gene that's important that's causing this entire set of defects in kids? Is it the whole region that's missing and that's why it's important? I think scientists believe that it's not the whole region. Every gene in that region is not causing these defects. And so using zebrafish, we can individually mutate each of these genes and then ask which one is contributing what part of that human phenomenon. And so this is an example of this powerful nature of these genetics. I'm not going to walk you through this slide, but what I want you to appreciate is that the history of zebrafish as a model organism is actually not that old. Streisinger was born before me, so I consider myself young. But he started playing with these fish, like I said, in the 60s. He died in 1984. And what's kind of truly remarkable about this model organism is that he was capable of training people in the short time that he was developing this model organism to continue it on. And the first R01 grant, or the first grant awarded by the National Institutes of Health using zebrafish in a model, was the early 90s. So this community started and worked with the fish for nearly 30 years before the federal government gave them any major funding. This was all done mostly by donors and private funds. In fact, Streisinger on the University of Oregon campus, which is where he was, kind of illegally used an old army barracks and built zebrafish racks in those buildings. They thought the buildings were unused. And that's where he did zebrafish for the first 25 years, where he started and then people finished. Currently, there are over 1,100 funded grants on zebrafish. So in just 30 years or so, right, you've had this huge explosion of these organisms. And then I think it's interesting. Streisinger started this, and one of the first postdocs he trained, her name is Judith Eisen. She's my scientific grandmother, because she trained my postdoctoral advisor, Bruce Apple. And so I do have a bit of a history, if you will, with the Streisinger scientific family. But during this time, right, these people, Judith and all of her counterparts during these early 20 years, really drove this model organism. And I think the splash that really kind of set this model organism on the map was a series of papers published in 1996. So I was a senior in high school, so it wasn't that long ago, because I'd like to think that I'm not that old. And what they did is they showed that this model organism can be used to study human disease very directly. You don't need to read these. But what they did is they did a mutant screen, where essentially they looked in wild populations of these zebrafish they had in a lab, and they screened for what we call phenotypes. They looked by eye. Do we see defects in any interesting organ system? And in this issue of Development in 1996, they published dozens of papers where they had identified mutants that affected things like pigment. So the cover shows you different stripe patterns of these fish. But beyond that, brain development, kidney development, heart development, every organ system. There was a paper and they had identified mutations that affected that organ system. So 30 years ago, the field basically hit with a bang. We have this model organism. It's a vertebrate. It's better than all the rest. And we now have all these amazing tools in terms of mutants that we can study to ask questions about human disease and development. And so in the last 30 years, we now have an International Zebrafish Society. I'm a card-carrying member. We also have an International Resource Center. So what that means is you can send your zebrafish to this resource center, and it allows you to share completely free to other people in the community. We have a Zebrafish Information Network, which is kind of like Zebrafish Twitter. It's where we all share techniques and protocols. There are courses now being taught all around the world to teach new trainees about zebrafish. This is one particular course at the Marine Biological Laboratory. It's in Woods Hole. I took this course 15 years ago. They're still teaching it every summer to new trainees. We now have disease model conferences. So not just meetings about the fish in general, but entire meetings surrounding specific diseases that zebrafish are used for to study human development. Meetings are all over the world, Asia, Europe, and currently, the best I could figure, there are about 1,200 labs in the world that use zebrafish as a model organism, which is more than, I think, 8,000 researchers. So that's, in 30 years, we've gone from one guy in the Army barracks at the University of Oregon to this model organism that's allowed us to study just about anything you would want to study that has to do with humans in terms of development and disease. And so I wanted to give you a slide now on just some of the things that people are using this for before I jump into the nervous system. So people are using zebrafish to study pediatric brain tumors. Okay? Not only can we create mutants where we can have them kind of spontaneously develop tumors in their brain, we can actually use these fish and transplant human tumor cells into a zebrafish brain and watch them develop tumors. They have stripes, they have melanin. They can spontaneously get melanoma. So people are using zebrafish to screen for drugs that can be used to treat melanoma. What's intriguing about zebrafish, like many fish and amphibians, is that they're incredibly regenerative. Okay? Humans, if we have a spinal cord injury, you have a skiing accident or you have a car accident, if it's bad enough, you can have a spinal cord injury that'll cause you to be paraplegic, for example. Zebrafish, however, you can completely cut their spinal cord in half, and they will completely regenerate it and be swimming again a month later. And so people are using these model organisms, particularly fish, to ask, how does that happen? What can we learn about the ways that the fish can drive this regeneration? And are there factors there, or things that are therapeutically targetable that we could try to create drugs for, that we could give to patients after they've had these kinds of accidents? People use fish to study cardiovascular development. Again, with humans, a lot of congenital heart defects you don't really see. Children are often, even with the ultrasound technologies we have now, it's very difficult to identify congenital heart defects. But with zebrafish, because they develop externally, we can label all of their vessels, that's all the stuff here in green, and their heart down here, and just watch development of the heart and screen for mutations that cause congenital heart defects and try to study them directly. Zebrafish have a pancreas. I know that sounds kind of crazy, but they do. You can give a fish type 2 diabetes. You feed it too much, it will get fat. It will get insulin resistance. But the kicker is you cannot give a fish type 1 diabetes. And what's intriguing about this is this goes along with their regenerative ability. They are so regenerative that you can completely ablate all of the beta cells, which is what happens. The beta cells die in a human with type 1 diabetes. They will completely regenerate these beta cells. You can make a fish type 2 diabetic, but you cannot make a type 1 diabetic. And so people are using these organisms to try to understand how are these beta cells regenerating? And are there ways that we can drive this regeneration in human patients with type 1 diabetes? Their fins regenerate, which I know doesn't look that exciting, but as a person maybe who loses a finger or a limb, learning how to potentially rebuild tissues that could lead to functionality again is something that's pretty incredible. And then finally, what people find most interesting, although I'm not a cardiologist, but it's cool, I guess, is that fish can regenerate their hearts. So what happens in humans when you have a heart attack is you lose blood flow, oxygen to portions of the muscle in your heart. Without that blood flow and that oxygen, that heart muscle will die. And so although you may survive, you're going to have a heart that's not functioning fully. And what happens is you end up with just kind of this fibrotic or scarred tissue, and this occurs in mice as well. But what's intriguing about zebrafish is we can actually remove 25% of the zebrafish heart, cut it out, and it'll completely regenerate in a month. And so again, it's this ability of this organism to regenerate all of these tissues that people are trying to exploit to try to come up with ways that we can drive these processes in humans. We're using them in small molecule screens. Almost every major pharmaceutical company in the world now uses zebrafish as its first platform. Previously, when pharmaceutical companies would screen, they often do it in an unbiased manner. They kind of create these libraries that have hundreds of thousands of small molecules. And they'll throw them into cell culture, or they'll try to give them to mice and look for molecules that look like they could be promising for certain human diseases. The problem with cell culture, as we talked about earlier, is it's way too simple. Anything that had been proven effective in cell culture was almost guaranteed to not be effective in an actual animal. And so now these companies are using zebrafish because you get thousands of eggs. You can put them in these high throughput plates, and you just put the drugs in each of these 96 wells, and you can screen by eye very, very rapidly. And so pharmaceutical companies are using this to identify what they call lead compounds, compounds that seem to be helpful for certain disease processes. They're also now being used even more for personalized medicine. So what we're learning, particularly in the cancer field, is that often when they take a biopsy or they remove a tumor from a resection, there's two ways people are using the zebrafish. One of the ways, as I mentioned, is they'll take those human cells and they'll actually transplant them into a live zebrafish. So this allows us to study how those cells behave in an organism where we could watch tumor formation. But even more excitingly, that's somebody's tumor. That's one patient's tumor. And the problem with a lot of cancer therapies is that they're very broad. They don't kind of hit every patient. And that's because all of the patients have different genetic mutations that are driving their cancer. But by modeling that human cancer in a fish, they can take those fish and then do a small molecule screen and identify drugs that are specific to help your tumor, and then rapidly give those patients those drugs. Now, this is all still very clinical trial, right? Because you're making a leap. But for many patients that have incurable cancers, the idea of having a fish at least help the doctor guess better as to which drug might lead to tumor death or at least halt its growth is incredibly powerful. But for me, and what I want to spend the last bit of the talk about is really using this model organism to study the nervous system. So what do these zebrafish have to offer? I've told you all of the great things that people are using them for, but I've held kind of the best for last, I think. So not only are they see-through, are they like humans, what we have are the molecular, biological, and genetic tricks where we can label cells in the nervous system in a live fish different colors. And what that allows us to do is label all of the cells in the nervous system a different color. So what you're looking at here is a fish that's both green and red. And so this here is the brain. The eye is here. I hope you can see that. And then this would be the spinal cord. And so you have cells that are green, cells that are red, and then some that express both green and red, so they appear yellow. What's great about this is it allows us to watch these cells, not just look at the cells kind of with a bright field without really seeing the structure of the cells, and I'll show you this in a second. And so zebrafish, this is a zebrafish nervous system. This is a human nervous system. We as zebrafish biologists often like to think that these humans are just fish that swim a little funny, because when you simply change the orientation, the cells are the same. They do the same thing. You have a brain. You have a spinal cord. Here's the human spinal cord. All of these red wisps are nerves. These are the parts of the nervous system that innervate your muscles and your organs. Okay? Humans have these exact same things. And so in my lab, we use these transgenic animals, and this is kind of why I think they're the most powerful for what we're interested in. This is a live zebrafish that we've mounted on a microscope, and we can watch in real time through the organism how these cells are behaving. And this is pretty powerful because never before have people really been able to look at the nervous system and ask, in all of its context, what are these cells doing and how do they behave in disease? And so you can imagine then coupling this with mutations or disease models or small molecules and starting to ask questions is incredibly powerful. And so my lab is particularly interested in unwrapping this idea of glial diversity in the vertebrate nervous system. And so I'm going to kind of define glia for you and kind of walk you through some of our projects. So in the nervous system, there are two main types of cells. There's neurons. These are often the cells that get kind of the most bang for your buck in the news, and that's because these are the cells that we consider electrically excitable. These are the cells that connect to one another and pass information and allow us to move, okay? And so this is just a series of pictures showing you the beautiful diversity of the types of neurons in the nervous system. All of these types of neurons exist in zebrafish, and they also exist in humans. The difference is that fish just have a fewer of them, right? They're not quite as complex as humans. But there's another cell type in the nervous system known as glia. So in your nervous system, in an adult nervous system, it's thought that there's about 100 billion neurons, okay? And to put that into perspective, this is what I tell the elementary school kids when I go out and do outreach. That's about the same number of stars in the Milky Way. That's kind of the assumption. So if you look up in the night sky, that's about how many neurons you have in your brain. Glia, which is the second type of cell type in your nervous system, you have 1 trillion of these cells. So you have 10 times the number of glia in your nervous system as you have neurons. But for the vast majority of neuroscience research, these cells have been ignored. And part of it, I think, has to do with their name. So glia was named because glia in Greek means glue. So when some of the original neuroanatomists were going through and kind of drawing pictures of the nervous system 100, 150 years ago, they saw all of these other cells that looked different from neurons. But because everything was packed so closely together, they assumed that these guys were just the cement of the nervous system. And so for 100 years, these cells were kind of left to the wayside because how could they possibly have been interesting for the nervous system? But what I'm going to tell you is that they're the most important cell type in your nervous system. But I also want you to appreciate just the beauty in terms of the diversity of these cell types, right? Just like neurons, where you see, you don't even have to know what kind of neurons these are, they all have a slightly different form. And in biology, if the form is different, then the function is different, right? If a cell looks different from its neighbor, it's probably doing something else. And the same goes true for the glia. And so when we think about neurons versus glia, right, this is kind of what's often, this is a friend of mine, he's an artist. This is kind of one of those memes, right, that kind of describes this. If you're a neuroscientist and you study neurons, you think neurons run the world, and the glia are simply the glue or the butler of the system, right? But if you're a glial biologist, we like to think that we're the puppeteers of the neurons. Now, in reality, this is probably correct, right? You need both cell types working optimally together. But what is true is that in a nervous system, you cannot exist without glia. So these cells, there are developmental disorders where these cells are missing or that they degenerate. And if they're gone, your nervous system goes crazy. So these cells are absolutely important. And so there are many types of glia. So when you think about your nervous system, your brain and your spinal cord here are the central nervous system. And then everything else that comes off is the peripheral nervous system. Okay? And it's peripheral because it controls all of your peripheral tissues. So locomotion, it controls your gut, it controls your heart rate. And when we take a snapshot from a textbook, this is a textbook that is currently used at UVA to teach neuroscience to our medical students, this is the picture you get describing glia. So you can appreciate my frustration when in fact this is it. And there's usually like a chapter out of like a 1,500-page book that describes these cells. This is what they call central nervous system glia, so brain and spinal cord, and here's peripheral nervous system. And so just in the last 9 years, since starting my lab here at UVA, we've discovered 2 additional types of glia. So that's just my lab in the last 9 years. And so my lab is particularly passionate about trying to really understand glial diversity, not just for diversity's sake, right? Not just because we like to discover cell types, but because I think it's fundamental for understanding human development and disease. And so my lab is really interested in how you build a nervous system and then how you rebuild it after injury. And again, glia. You guys are going to leave. My daughter knows, actually, that glia is the most important cell type in the nervous system. All of Woodbrook Elementary in Albemarle County also knows. And so in my lab, we use many of these same techniques that I've been telling you about zebrafish. We use small molecule screening to identify compounds that affect glia. We couple this with in vivo imaging. Again, we can live mount these fish on pretty expensive microscopes, but watch in an intact system how these cells behave. We now have this fancy genetic technique known as CRISPR. Many of you may be hearing about this on the news. It's basically a very rapid way to create a mutation in any organism, including humans. You can not only create mutations, you can fix mutations. And so this technology, coupled with imaging and drug screening, really gives us what I think, I'm biased, I know, the most powerful model organism to study the nervous system. And then finally, we can also shoot lasers, which the lab appreciates. It's kind of cool. But what it allows us to do is not just affect the whole nervous system. We can actually go in, and instead of creating a mutation that affects every cell in your brain, we can create a mutation and only have that mutation occur in a single cell because we can control with a laser where that cell, and which cell has a mutation. And so it allows us to very closely mimic human disease. And so I want to tell you a few vignettes today about just some of the stuff we're doing in the lab. The first is the importance of crossable borders. Okay? And I realize we're after the election. What are nervous system borders? So when you think about your nervous system, I described to you your central nervous system and your peripheral nervous system. And they're described that way because they are, in fact, 2 separate nervous systems. So if we were to take a cross-section through a human, straight through the middle, and fillet it back up, what you get is this oversimplified diagram. But this oval here is your spinal cord. Okay? And so everything in the oval is your central nervous system. So like this motor neuron, for example. Everything outside of this oval is your peripheral nervous system. Now what's important about these borders is that you have to have stuff in your peripheral nervous system and stuff in your central nervous system communicating. You need to be able to have sensory information, for example, I realize this is quite small, that if you put your hand on a hot plate, the axons and the neurons that are detecting that are peripheral. But they don't move anything, they're simply receiving environmental information. That information has to be shuttled into the central nervous system where it's processed, and your body then initiates activity of motor neurons that says, Sarah, get your hand off that stove. And so what's essential and important about these borders is that you have information that can cross through them, and it can cross through it very rapidly so that you get this integrated response. And the example I gave you was very basic, right? This idea of a hot plate, move your hand. But everything we do, both consciously and subconsciously, requires this constant information flow. So these borders exist, but nobody's really studied them. And so I, being someone who just likes to study weird things, became fascinated with these borders, and in particular glial cells. So the reason this happened, and I know this slide is a little bit busy, so I'm going to simplify it, is the first cell type that I discovered as a postdoctoral fellow. It's a cell type known as perineural glia. I tried to call it Kucenas glia, but my boss wouldn't let me. So apparently the days of naming things after yourself ended. But perineural glia, so this is that same spinal cord, but early in development are these green circles. And what I discovered is during development, these circles, these actual glia, migrated across these boundaries. They're supposed to move between your central nervous system and peripheral nervous system. This is something that nobody appreciated. Everybody thought that these borders were just there and they should be left alone and something magical happens. But what we discovered is that glia actually go back and forth, and that it creates a normal nervous system at the end of these developmental processes. But I think what's most important is it's not just that these borders exist, but that this crossing of cells across them is essential. You have to have semi-permeable borders. You need movement of cells in order to have a normal nervous system. So here's a normal nervous system. It's from the last slide where you see these green cells moving across this border. If we perturb the movement of these cells in a couple different ways, hopefully what you can appreciate is that these two diagrams don't look like this diagram. If you don't allow the passage of cells, and you do not get normal nervous system development. And what's important is you don't just disrupt the peripheral nervous system or the central nervous system. Both portions of your nervous system are disrupted. So this movement is essential. And so one of the questions that we've been wrestling with for 9 years in my lab now is how are these borders semi-permeable? How does the nervous system know to let some cells out, like those perineural glia I just described to you, while not letting other cells out? Because you don't want all of your cells crossing back and forth all of the time. There's a reason that there's a bit of a segregation. And so the short story is the identification of a second new glial cell type, known as motor exit point glia. And so hopefully you can appreciate in 9 years, 2 new glial cell types. That builds that textbook by 50%, that picture, right? So, and we're not even looking. We're just asking interesting biological questions. But what we discovered is that these cells, motor exit point glia, are these green cells here in this diagram. They actually sit at that border. But what's intriguing about them is that they're not just keeping everybody out. They're very selective. They allow certain cells to pass through and not other cells. And we can test this in fish by using a laser. Actually, let me start up. What you're going to see is a movie. I realize this is a little bit fast, where these arrows, believe it or not, are pointing towards those motor exit point glia. We can use a laser and we can kill just those border cells. And we're going to see what happens. And hopefully you can appreciate the cells, boom, have just been killed. And immediately after you remove those border cells, we can get cells from your spinal cord out into your peripheral nervous system. Okay? So this suggested to us that there's very dynamic regulation at these border cells, and that this semi-permeable nature of your nervous system is essential. Because these cells normally, in a normal human, in a normal fish, you don't want them out in your peripheral nervous system. So the next question we asked as scientists was, well, how do they do it? Right? It's never good enough to just show that a cell type did something. The question is, what are the genes that drive these processes? And so what we did is go back to what zebrafish are really great for and do small molecule screening. And what we did is we screened 3,000 compounds. This was done by an undergraduate in the lab in a summer. And that's phenomenal, right? That they could take 3,000 compounds and in just 3 months screen and identify 10 out of that 3,000 where we found drugs that cause cells to move across that border inappropriately. And so here's an example where we've discovered a small molecule that allows a very specific subtype of glia to cross this boundary. So if we treat this zebrafish, hopefully what you can appreciate is that that yellow cell is supposed to stay up here in the spinal cord. But what it did with the drug treatment is actually cross that border into the peripheral nervous system. Now, we didn't just do this just because we were interested in what causes that crossing. We also want to know, can good come from crossing? Can we promote crossing of glial cells across those borders? And is that therapeutically tractable? Is there a reason why a human with a disease might need a cell type to move across those borders to aid in regeneration. And one of the diseases we're particularly interested in the lab is Charcot-Marie tooth disease. It's kind of a class of muscular dystrophies. These patients, it's considered a muscular dystrophy because the muscle degenerates. But the reason the muscle degenerates is because you have defects in the peripheral glia. So your glia gets sick in the peripheral nervous system. And if that happens and goes unchecked, your muscle starts to degenerate, and then you lose mobility. But what's intriguing about these patients is all of their central glia are normal. They're unaffected in these diseases. And so what we thought is, can we actually induce a little bit of that border crossing by these spinal cord glia? And if we can they repair those defects? And so can we kind of take that glial migration and manipulate it to treat disease? And so what we used is a model in fish of a peripheral neuropathy. So I told you fish are great models. There's basically a model for almost every disease. They get peripheral neuropathies just like humans do. They can be caused by injury, right? But also I told you can give a fish type 2 diabetes. And so the fish, if given type 2 diabetes, will also get diabetic neuropathy, where you start to lose the glia, which are labeled here in green, along your peripheral nerves in the mutant. And so what you're left with is a fish that doesn't swim well, right? It's kind of very lethargic, and it will die early. And so we wanted to treat these fish with that small molecule and ask, does border crossing with those glial cells promote repair? And so this is a mutant fish that we've treated with our small molecule. And lo and behold, we can get those central glia to cross that border and come out into the peripheral nervous system. But what's most exciting is that not only can they cross into the part of the nervous system they're normally restricted from entering, they're actually incredibly beneficial for disease. So in our control mutant fish, you can see that the glial cells, which would be here if they were there, marked by this green arrow, are completely absent. But in the fish that we've treated with that small molecule, we can promote those central glia to come out and cross that border and then fix all of those issues in the peripheral nervous system. And these zebrafish swim better, they breed better, and they live longer. And so we're very excited about using these kind of small molecules in zebrafish to ask questions about treating disease in novel ways. And now I'm going to transition into another project in the lab where we're really interested in multiple sclerosis. So MS is an autoimmune disease that affects glia, primarily in your nervous system. And so when we think about your brain and spinal cord, we talked about all of those neurons that create connections with one another. But the reason that we're able to think so quickly and move so quickly is that you have this basically electrical tape wrapped around almost all of your axons, just like electrical tape on electrical wires, that facilitates really rapid conduction of information. And this stuff here in these little rectangles is made by glia, and it's called myelin, okay? And this myelin is what's attacked in disorders like multiple sclerosis. And so what ends up happening is you have neurons that are myelinated by glia, which have these white sheaths. But in MS, the immune system inappropriately attacks that myelin and essentially chews it off your axons. And what you're left with is a series of symptoms. Now, MS is relapsing remitting, which is particularly frustrating for patients because they'll go through bouts of disability where the myelin has been taken off of the axon. And so depending on where the lesion is, where the myelin has been lost, you can have issues with vision, some of your senses, your urinary system. Eventually, this becomes a progressive disorder where you lose all of the myelin in your nervous system, and then you die. And so we're very interested in using zebrafish as a model to start to understand this disease. And so what we did, and by we I mean a really talented graduate student, none of this is work I did, is she created a model of multiple sclerosis in zebrafish. So there are lots of models of MS. Mice, you can give MS to, right? That's kind of that canonical model. But the problem is you can't see through it. You can't see what's happening to the myelin. And so what Angie, a graduate student, did is she initiated a demyelinating event in zebrafish. So you're looking here at the spinal cord. And what these arrows are pointing to in this side, this is the unannotated movie, is that we can get breakdown and blebbing of myelin. We can create a demyelinating scenario in zebrafish. So this is very exciting for one hand because nobody's ever seen demyelination before in vivo. So we now have the ability to watch how myelin falls off of axons during MS. But then we can couple this with small molecule screening, I feel like a record, right? Where we can screen for molecules that don't let this happen, that block that demyelinating event. And in fact, we've discovered a small molecule that prevents that demyelination. And so you'll have to believe me, but this fish has been induced to have a demyelinating lesion. But because it's being treated by this small molecule, you don't see that kind of breakdown or that blebbing of myelin. And so zebrafish in our lab is really allowing us to drive these projects forward to really ask questions about therapeutics, particularly geared at glia. I have two small little stories at the end. I mentioned this in the past. You saw my frustration, why glial diversity is so important. I think part of it is I don't like that diagram that shows you 6 glia. You have 100 trillion of these cells in your body. There's probably not just 6 types, right? And so it's also important and interesting when you think about evolution of the nervous system and glia. Okay, here's another one of these trees. The first kind of picture, or the first kind of acquisition of glia is way down here with like a sea slug, right? So you have to think to yourself, a lot of people think, well, I'm not like a sea slug, right? I'm very, very different. But in fact, your glia evolved from an organism that attained these cells at this stage in development. Now, they're poorly developed. They're the rudiments of what glia are. But they did the exact same thing that the glia in your brain do. As evolution has occurred and we've kind of gotten speciation and more diversification, you can start to see these species that have glia as well. And you're probably thinking to yourself, I don't look a whole lot like those species. But believe it or not, your nervous system is very similar. The same cells are there. The same genes that are driving their development are there, right? But what has happened is you are up here. So something has happened that has caused your nervous system to function in a different way than all of these other organisms. Now, again, I'm biased. I'm a glial biologist. But there's only one way to make a neuron. All neurons in life look the same, right? A neuron down here is very, very similar structurally to a neuron up here in our brains. It's the glial cells that have completely changed their form and therefore their function. And they've really exploded in terms of diversity. And many people think that this is the reason that we have things like cognition and other species don't. And so if we think about these glial functions, again, this is one of those slides that angers me. Here are those glial cell types, and it's not because my cells are missing. It's just because this is an oversimplification, right? But they give you these very basic functions of these cells. Now, in a worm, those ependymal cells are creating barriers, just like they are in your brain. So this slide doesn't tell us anything about the diversification and why human brain develops the way it does and why we get diseases that other organisms don't. And this diversification, again, is why we're here. This is Amphioxus. It's our ancestor. It's the very first vertebrate. So the reason you are walking upright is because this species developed a notochord. So you can thank this species. But the reason that we are very, very different is this cell type. It's a glial cell type known as astrocytes. They have astrocytes. We have astrocytes. But the difference is we have probably 1,000 more fold, and they look very different. Okay, they behave different. And this is kind of illustrated here. Now, this is a mouse and human, but B is a mouse astrocyte. So this is a label of one of those glial cells. A is a human astrocyte. And so even though these cell types in your textbook would do the same thing, you can look and see that they look very, very different. And in fact, the complexity of this human astrocyte is why we think human brains develop the way they do. And these cells are implicated in things like schizophrenia, obsessive compulsive disorder, autism. And it's very difficult to model things like schizophrenia in a mouse. And many people are now appreciating we're trying to model a disease in an organism that doesn't have the complexity of the cell types that allow us to model it. Zebrafish, believe it or not, have cells that look a whole lot more like a human astrocyte than mice do. And so in my lab, we're chasing glial diversity, kind of with a sledgehammer. What we do is we use zebrafish. And because I showed you all of those labels where we can label these cells different colors, we can basically chew up the fish, not literally, but in a mortar and pestle, and isolate only the glia out of these guys. And then what we can do is do genomics. We can ask what every glial cell type expresses and compare it to what we know about all other glia. And so what we're left with, and I don't have any more data really to tell you about this, is what we call the Kucenas lab magic file, where through some computational processing that we're doing, we're starting to discover almost weekly markers that seem to label new glial cell types in the vertebrate nervous system, which is incredibly powerful for us. So this is what the textbook UVA med students are currently learning about glia. Textbooks are edited every 10 to 15 years. I appreciate that. But I hope that before I retire, I don't need my name in the textbook, but I would like a cartoon of these other glia. right? And what we're finding is that they're incredibly important in disease. And so here's just one example of a gene that labels a specific subtype of glia, CRIP3. What you're going to see is a row of glia here in the spinal cord. The spinal cord is this whole picture. There's a very select population of glia that turn on this gene and then turn it off within a day of development. They express no other known marker of glial cells that we have in zebrafish. And so we're very, very excited to be able to use some of this data to not only identify these novel cell types, but then ask, what are they doing? And what does that tell us about human development? And then the last little vignette is, I can't not talk about cancer because glial cells are incredibly proliferative. You have 1 trillion of them in your brain, okay? And the nature of these cells is to act very dynamically during human life, to clear the waste out of your brain. They help you make memories. They clear debris. They modulate blood flow. So unlike other cells in your body that just kind of differentiate and become mature and don't do much, these glial cells are constantly working. They're constantly being attacked, if you will. And they're very proliferative. And what ends up happening is you can get diseases like glioblastoma. And John McCain, this is the disorder that he died of. The average life expectancy of someone with glioblastoma is 14 months. This is a highly aggressive glial cancer. And what I want you to appreciate is that glial cells are not supposed to form these tumors, okay? And this is a question we're very interested in understanding. What causes a cell type that would normally not kind of aggregate into a tumor form a tumor? And so what we can do is use in vivo imaging in zebrafish and ask, how do these glial cells normally space? During normal and physiological conditions, how do they space from one another? And how do they stay spaced? Now, this is a very kind of oversimplified movie because this is actually the amount of process activity that these glial cells have in vivo. These are all glial cells labeled here in black, but what I want you to appreciate is they all have these dozens of processes that are constantly moving back and forth and bouncing off of one another. So this glial spacing isn't as simple as you're born, you migrate, and you sit still. These glial cells are constantly interacting and pushing each other around so that they occupy their own space. And so what we want to do is use the zebrafish to understand how do you get normal spacing. And then we have a zebrafish model of glioblastoma and ask how are those kind of events that allow normal spacing to kind of go awry so that you get tumor formation. And so I just want to end. I went a little bit over. I apologize. I get excited about zebrafish and model organisms and neuroscience. What I hopefully kind of imparted to you is that zebrafish are contributing to biomedical research in ways that I think people don't appreciate, and I think we're just at the cusp of really being able to use this model organism to understand human development and disease. I specifically told you, if you don't remember anything else, this is what I tell my daughter, glia are the most important cell type in your brain, and you should thank them for everything that you have going on for you. And I told you a couple stories about borders and multiple sclerosis, et cetera. There's a lot of stuff my lab is interested in. I didn't tell you everything just because I didn't want you all to fall asleep. But these are the kind of the basic things that we're very, very interested in. And so with that, these are the people that I am lucky enough to get to train and work with in the lab. We have both graduate students, postdocs, and undergraduates, which I think is actually a really powerful way to run a research laboratory. We've had some really fantastic, generous funding. So with that, I would love to take any questions. Yeah.
So what are the small molecules?
Okay.
Are they currently thought to be drugs that are relevant for humans, or is this a completely new thing?
Yeah, so there's, I haven't been able to tell you, I couldn't tell you all of it because some of it is with the UVA patent office, right? Some of them are small molecules that are out of some of these kind of 100,000 molecule library, and what they are is just simply been proven to be bioactive. So some of them we don't know what they do yet. But the one in terms of moving cells across a crossable border, that's actually already an FDA-approved drug. And so we're very excited. We're looking for pharmaceutical partners because I don't want to do mouse work because you can't see through the mice. So I would love to have a pharmaceutical, also to pay for it, to ask questions about if we have some of these same neuropathy models in mouse, if we give these compounds, do we see the same migration of cells and is it helpful? Because that would be the next step, right, for looking at a human treatment. Yep.
Other model organisms being explored as we speak. I don't you hear things about jellyfish for memory and whatever else, but what else is being looked at?
Yes, so the question was model organisms. What else is being looked at? Basically everything you can imagine. I saw a recent article about the NSF and a couple other major funding agencies worldwide would like to sequence, quote unquote, all the things. So basically every organism on Earth they'd like to sequence because understanding diversity, right, from a very basic standpoint. But also, are there other model organisms that people aren't thinking about that may be more similar to humans than others? So jellyfish, actually there was a beautiful study done on squid recently in Current Biology, and octopus, actually, where octopuses, I think is the correct word, are incredibly intelligent. And what they found is they were able to teach an octopus in a separate tank a task to open a box. Its neighbor was sitting in its tank, was not taught the task, but could learn from watching the first one. And so kind of developing some of these organisms that may seem not at all like us, but actually have these behaviors that would allow us to ask questions about how we learn and memory. That same paper, actually, octopuses can get highly addicted to methamphetamines, and so they also want to use it as a model organism for addiction to try to understand some of these phenomena that are just very, very difficult to untangle in like a human brain. Yeah?
So if you try not to ask the why question, because it is not science.
It's hard to, yeah.
If you turn on the ability to regenerate in an organism like the humans, what do you think the trade-off is? Or what do you think is you will lose or inhibit or?
Right, so the question is, what might happen, good or bad, if you drive a regenerative program in humans? I think that's a fantastic question. We know from things like the use of stem cells. So a lot of people have thought, well, if we can't drive regeneration, what if we put in stem cells, right? These cells that are multipotent that might be able to rebuild the system on their own. So in that case, what we're finding, and there was a study done in California with spinal cord injury patients actually, they got tumors. So driving regeneration and driving some of these developmental processes, actually, you want to be careful of in an adult system. I mean, development is incredibly tightly regulated. And so these glial cells during development that are bouncing off of one another and trying to space, there's a lot of programs and cues there to keep everybody in place. But we know that those things disappear in an adult nervous system. And so you worry that by trying to promote this remigration in an adult system where it doesn't have those checks and balances, you could end up very quickly with a bad event. Yeah. But we still like to think that there's power there. If we could figure out developmentally, right, and as a developmental biologist, that's what I am at heart, if we could figure out how you build the system and all of those layers of checks and balances, and then identify small molecules or other mechanisms to drive these events, plus add back those things that help keep the system in check, I think that could be incredibly powerful.
That was my question.
Oh, okay, yep.
All right.
Probably
Great.
a question all of us didn't think to ask.
Thank you so much.
Thank you so much, Sarah.